Hi everyone, welcome back to Useful Genetics. This is lecture 3B, where we're going to talk about protein folding and function. We'll talk about how proteins fold into complex functional structures with what biochemists distinguish as 4 different levels of structure. We'll talk about proteins whose job is to help keep proteins out of trouble while they're still sort of new and haven't yet achieved their proper folded structure. And we'll talk about the roles of proteins in the cell. On the right here is a simulated protein. It's a very short peptide. You can see these are different side chains along the chain. And this is an animation simulating the actual protein, folding of the protein. And you'll see there's a lot of random motion driven by random thermal energy in the cell that eventually brings compatible parts of the protein chain together to fold. 2 points about what makes parts of the protein compatible. Some parts of the protein, some amino acid side chains, have the property of being hydrophilic. They like water. And this causes them to stay on the outside of the folded protein, where they can interact with the water molecules in the aqueous environment of the cell's cytoplasm. Other side chains are hydrophobic. They avoid water, and they tend to fold into structures where they're together, sheltered from the water on the outside of the cell. So they tend to be on the inside of the folded protein. Now that's a very oversimplification, but it's a fundamental principle nevertheless. Now, biochemists distinguish 4 levels of folding in proteins, although 2 of them aren't really folding at all. The first level, which isn't really folding at all, is just the sequence of the amino acids in the protein chain. The second level is a kind of local folding, which actually is easier to understand if we look at it in the third structure. So here's a segment where part of the protein has formed a structure called an alpha helix. And this is driven largely by just the structure of the backbone. It's partially independent of the properties of the particular amino acid side chains. Here's a different kind of local structure called a beta sheet. Here's a part of that protein that has folded into a beta sheet. The third level of structure, which is really the main sequence-driven level of structure, is the complex folding of the protein back onto itself in many different ways, so that all of these different parts of the protein are interacting with each other by various chemical interactions, usually not covalent interactions, ionic interactions, hydrophobic and hydrophilic interactions, that fold the protein into its particular shape. The fourth level, which again is not really folding, is the assembly of different protein subunits, the products of different genes, into a larger sort of megaprotein, which consists of different subunits from different genes. You encountered an example of this, although you might not have realized it, in the last module when we talked about the alpha globin and beta globin genes. These are genes on different chromosomes. They code for related but different proteins. But the 2 proteins, the alpha and the beta globin, assemble together into the 4-part protein called hemoglobin, which actually carries the oxygen in our blood. Now, for protein folding, it's like folding a map. There's a lot more ways to do it wrong than there are ways to do it right. And the larger a protein is, the longer the primary sequence is, the more likely the protein is to run into trouble during the folding process, because most proteins are much more complex than that simple one in the animation. And so cells have special other proteins, kind of like metaproteins, whose job is to help proteins fold up. And these proteins are called, cleverly, chaperonins. Here's an example of a chaperonin complex. It's actually made of many chaperonin subunits, here's what it looks like seen from the end. It's sort of a hollow bag in which proteins, young new proteins that haven't yet achieved their mature structure, have a sheltered environment where they can explore interactions while the chaperone, it's really like a chaperone at a party, the chaperone proteins sort of promote appropriate interactions and gently discourage inappropriate interactions. So here's a question for you. How come proteins can fold up so many different ways when different DNAs always forms the same double helix?